Um, Thanks for coming to the lecture, the next lecture in the CPBF Summer School on the Physics of Life. So my lecture is on the topic, how the brain interprets complex signals. So let me introduce myself. My name is Vijay Balasubramanian. I'm the Department of Physics and with the Computational Neuroscience Initiative at the University of Pennsylvania. Wonderful. So let's see. So the topic of this lecture is this thing, which is inside your head, and the brain is the object in your body that gathers information from the world, makes decisions for the future, learns from experience, and tries to remember things. Right. So this is what all animals rely on in order to be able to act in the world. Now, the challenge faced by the brain is that, it's a very limited object with a certain number of neurons and a certain number of connections. But the world is very big, very complex, and often unpredictable. And we have strictly limited resources with which to process the world. So the basic question, indeed, in the entire field of neuroscience, you might say, is what are the organizational principles that allow neural circuits to meet the enormous challenge of dealing with the world with such a limited computational device. So of course, that is the subject of the field of neuroscience as a whole, and there's an enormous number of neuroscientists, excellent people, all great experimentalists, wonderful theorists, etc. So you could ask the question, what do physicists bring to the table in this quest? Why are we needed at all? Well, so in my view, we bring various new things to the table. So one thing is we bring new kinds of technique, both theoretical and experimental techniques, many of which have been showcased in the school so far. So we bring techniques that the biologists don't necessarily have. But I think the most important thing that we bring out of physics is a belief in the existence of principles or explanatory frameworks. So basically, in physics, we have a tendency to believe that there's an answer to the question why, as in, why does this particle recoil when that particle hits it? Why is momentum conserved? Why is there this set of particles in the world? And we try to answer these questions about the principles that govern the organization of systems. Whereas it tends to be in biology, the biologists focus on the questions what is happening and how it's happening, just because of the enormous diversity of phenomena in the living world. But as physicists, we bring this sort of distinctive approach, asking why are things happening, and try to answer that question. And I will try to illustrate today in the domain of neuroscience how we apply this kind of approach to the organization of neural circuits. Now, few of you, I think, are actually well-versed in neuroscience. So I'm going to start out by discussing the structure and architecture of circuits in the brain. So I think the previous lecture in this series also talked a little bit about this. So I'm going to try to cover slightly orthogonal material or reinforce that material because seeing things twice helps understand things better. So first of all, what's the brain made of? The brain is made of neurons. Neurons are specialized cells. And like all cells, cells have cell bodies and do the usual, and a nucleus and carry out the usual cellular functions. But neurons are specialized cells whose job is to compute and communicate. So as such, there's a cell body, and attached to the cell body, there are a whole bunch of dendrites. The dendrites are the input wires of the neuron. And the dendrites then take in information from presynaptic neurons. They then produce some kind of signal. The signal is passed along the axon. The axon carries a message down to the end. And at the end, the axon sort of branches out into these axon terminals, which then communicate through chemical synapses onto the next neuron. And in this way, a network of neuron gets built up. Now, neurons are essentially electrical devices. So they have some baseline potential of about minus 70 millivolts. And when ions flow in, because you've had a communication from outside, the potential increases. And when it reaches some threshold value, there's a sort of runaway feedback process that causes this to shoot up and then shoot back down again in a very, very sharp localized pulse. So this is an expanded version. And if you view it within less than a millisecond, then you get this very sharp upward pulse that goes up and then comes back down. This is called an action potential or a spike. And it's essentially a digital signal that a neuron can send to another neuron. So basically, the neuron is bopping along at minus 70 millivolts, and then every once in a while it goes boom, boom, boom. And those booms or spikes or action potentials are the digital messages that one neuron can send to another. So the task of understanding neural computation is to understand why and how these kinds of digital signals get produced, how they're communicated, and how they're interpreted by other neurons in the brain. Now, this discovery that neurons basically function through electricity, and really you're talking about electrical signals and how they're propagated, goes back a long way, a couple of hundred years actually, because soon after, people started discovering and understanding things about electricity and magnetism, people made things like batteries, and someone surely had the bright idea of sticking, the current from some electrical cell into, let's say, a frog's leg. And if you do that, you'll find that the frog's leg, a dead frog's leg, will kick. And that's because the electrical potential is driving the neuron to fire. And if a neuron in the leg, in the muscles fires, it'll make the leg kick. In fact, this was so impressive to people at the time that for a while people even thought that electricity was really what life was made of. And people would do stuff like at the Royal Society in England, somebody took the body of an executed criminal and stuffed it full of electricity, and the body sort of goes around like this, and they thought, oh my God, I've revitalized this dead human. That's of course not the case, it's just the muscles moving around. But that's what led to things like the book Frankenstein by Mary Shelley and others, this idea originally from the idea of revitalizing dead things by sort of pumping them full of electricity. In any case, for our purposes, the key point is that neurons are little electrical circuit elements, and our goal is to understand how these things produce the kinds of behaviors we care about. Now, of course, single neurons by themselves can't produce, can't experience love, can't interpret poetry, can't produce art, and so on. So most of the things we care about in humans arise because, let me see, there's a couple of questions here. According to one of the questions, I'm not visible, so I wonder how I make myself visible. Give me a moment here, and I'm going to try to make myself visible to video. I'm supposed to be visible. Okay, so some people can see me and some people can't see me. Okay. There's a question that says, oh, thanks very much. The questions are being answered and various people are saying they can see me, so we're good. But there's a question from Andrew Graham saying, what part of the action potential in the neuron relates to the actual information being transmitted? So there's often some debate about this kind of question, but really you can think about this as saying that if there's the action potential, it's one very stereotyped large pulse. This thing moves to the end of the neuron and triggers the release of neurotransmitter at the axon terminal. So it isn't a part of the action potential that carries the actual information. It's the presence or absence. It's like a digital signal. If it's present, it's carried a little bit of information. And if it's absent, then that information has gone away. I hope that answers the question. Anyway, so that's how the neuron works. And neurons are put together into gigantic networks, which collectively do all the processing. So I'm going to start talking about these networks at the largest scale, and then I'm going to come down to the smallest scale. So at the largest scale, all the neurons assemble themselves into the whole brain, and the whole brain is divided up into many areas. So for example, here is the frontal lobe, here is the occipital lobe, and in the occipital lobe are localized the circuits that deal with vision. So this is why if you bang the back of your head, if you fall and bang the back of your head, you see stars and your vision is blurry. And that's essentially because you have a bruise in the circuits, if you like, that do vision, and so they're not working as well. And the front of the head here, there are the circuits that do things like planning, personality, and stuff. This is why you should avoid having a lobotomy. A lobotomy used to be a treatment which involves sort of basically scrambling the frontal lobe over there. It'll completely destroy your personality because the circuits there sort of control a lot of what we value in human behavior and the ability to plan and things like that. So the idea that overall function was localized in this way actually goes back a long way. There's a papyrus from some building site in ancient Egypt, in which the author describes how sometimes the workers on the site get bashed in the head by something. And when they do, it's funny, he says, that it's not like they completely lose all their functions. If you bash them in one place, they lose the ability to talk. In another place, the left hand stops working, but the right hand still works. So there's a proposal in this ancient papyrus, in this manuscript, suggesting that the different functions that make up animal behavior are localized in different parts of the brain. So this is perhaps the earliest such record of that idea. And then in the 19th century, there was a French doctor, Broca, who had a patient, and there was a nail or something that pierced the head of one of his patients in a particular location. So it was actually this area over here, Broca's area as it's now called. And this patient stopped being able to produce speech, although they continued to understand speech in the usual way. So starting with that discovery of Broca in modern times, there has been an expansion of the idea that different kinds of functions are localized in the brain. That's because the circuits in different regions of the brain are actually localized in different, the circuits in different regions of the brain carry out specialized functions one way or the other. One way of trying to understand this better is to start focusing in closer and closer and closer into the circuit. So let's take this region, the motor control region, and focus in on that. So here it is. So this is that little strip. That's a strip running down the side of your head here, and the side of your head here. I hope you can see my hands, right? And along that strip, this is the region of the brain that is the motor cortex, and you'll find that within it there's a still further specialization of function. So for example, the circuits at the top there control the knee, and the circuits on the side here control the various muscles in the face. This is why sometimes if you see a person who has a stroke, so they have some stroke over here, then they'll find that the left part of the face sags, but the right side doesn't. That's because the circuits in this region have been damaged. There are a few questions here. So one question says, what sets the threshold for the action potential? Is it neurotransmitter dependent? So I think the question being asked by Kevin Scritters has to do with the threshold at which, once you've changed the potential, it shoots up and shoots back down. So that's not set by neurotransmitters. It's set by the number of ion channels in the cell membrane and how they interact. Actually, this is part of the very famous work of Hodgkin and Huxley. The first Nobel Prize in biophysics went to Hodgkin and Huxley for discovering the set of equations that control the nonlinear self-interaction of the voltage-dependent ion channels in the surface of a neuron, and that's what sets the threshold for that's how it sets the threshold for the action potential. Saad has asked a question from the asking whether suggesting that the way we know about this localization of function is by looking at brain activity like MRI. And indeed, it's true that nowadays we have imaging techniques like magnetic resonance imaging that can tell you which bits of the brain are active when you're doing different things. But he asks, how can you be sure that there isn't just a coincidental correlation that when you see this bit of the brain light up while you're talking, that indeed it's this bit of the brain that's responsible? That's a very good point, that you need to do more than correlation. And indeed, there are many sort of causal tests that have occurred. Either they have people who have, have brain damage in one side or the other. There are accidents, this, that, the other. You can also do things like before doing neurosurgery, sometimes you've got to test which bit of the brain does what. So you take a electrode and you lightly stimulate different parts of the brain to see what happens. And you will see that if you stimulate different parts of the brain, it produces different kinds of activity. So those are the pieces of evidence that we have for why function is localized in this way. So then you can take a piece of the brain like this and then focus in still further. So I'm going to take one of my favorite bits of the brain, the retina. And the retina, here's a very famous drawing of the retina drawn by Ramon y Cajal, basically the founder of modern neuroscience, if you like. And he drew these beautiful, beautiful pictures after doing very careful anatomical studies. And this is a diagram he drew of the retina. So over here, so this is, a little piece of the brain that's stuck out in front of your head. It's really a piece of your central brain that's been put in a stalk and put in front of your head so that you can see easily. And this piece of the brain is tasked with the function of taking light and converting it into electrical signals. So here at the one end, you see here the rods and cones, which convert light into electricity. they produce an analog signal. At the second layer here, there are the so-called bipolar cells, and the bipolar cells, do some kind of analog computation with the signals from the photoreceptors. And the third layer here produces are the so-called ganglion cells, and the ganglion cells basically do some various computations with the signal coming out of the bipolar cells and essentially detect visual features. So some of these ganglion cells are responsible for detecting bright spots. Those are called on cells. Some detect dark spots. Those are called off cells. Some detect left to right motion. Others detect right to left motion, up down motion. There's a whole plethora of these kinds of feature detectors. So this picture drawn by Causal is your canonical, is the thing that canonically gives rise to the idea of the neural network. You have a network of multiple different kinds of neurons, here organized in 3 layers. So this is a 3-layer neural network, and the language that we would now use these days when we build artificial neural networks. The 3 layers do somewhat different things. They all branch out and gather information from each other. And at the end of the day, this particular neural network does something that's very similar to what many machine vision algorithms do, which is to say, at the end of the day, it takes the visual scene and breaks it up into different kinds of visual features. As I said, bright spot, dark spots, left to right motion, color, and so on, and sends the signals out along these long axons. And these long axons coming out of the retina form a bundle, which is the optic nerve. So this is the structure of the retina. So even this is a little bit simplistic, actually, because using modern techniques, which Ramon y Cajal didn't have access to 100 years ago, you can show that actually it's much more detailed than this. There are 3 cones and 1 rod type, and that feeds into 10 different types of bipolar cell types. That feeds into 15, or actually now the number is 20. This was in 2001. There's about 20 different kinds of retinal ganglion cell types, these are the feature detectors of the early visual system. And what's more, running sideways. So you remember, we just said that there's a feed-forward neural network. The photoreceptors feed to the bipolar cells, which feed to the ganglion cells to extract the visual features. But in the sideways direction, in the lateral direction, there are other kinds of neurons. There are these so-called interneurons. Here are the horizontal cells. And here there are the so-called amacrine cells. And these different types of cells all, as you can see, look very different. These are sort of schematic pictures of what they look. And there's a rule in biology that form follows function. So things that look different do different. So we can expect these things to perform different computational functions. And indeed, these amacrine cells and these horizontal cells, what they do is they are inhibitory neurons. So what they do is they actually, the feed-forward neurons take incoming information, process it, and send it forward. And these sideways, these lateral interneurons, prune away some of that information, they cut it away. So one way of thinking about this is as follows. So somebody once asked Michelangelo how he managed to sculpt this great sculpture of David, which is sitting around these days in Florence. And he answered, well, actually David was already the marble. My job was to remove everything that was irrelevant, right? All the extra stuff. So these amacrine cells and these horizontal cells have that job. In comes the whole visual stream. There's a huge amount of information in it. Most of it is irrelevant to animal behavior. So the computation required to prune away what's irrelevant is basically carried out by this huge number of cell types. So you see, the retina alone, a tiny, tiny, tiny piece of the brain, already has 60 different types of cells. And you can view these 60 types of cells as the kind of computational repertoire of the eye, of the retina. So in particular, if you built a silicon circuit, you would think about it in terms of resistors and diodes and capacitors and transistors and what have you. There's this repertoire of circuit elements that you string together to carry out the computation. And what you're seeing here is the repertoire of computational elements. And there's a real sense in which this is like a silicon circuit, because you see, if you look in any vertebrate retina, you will find that the slow bipolar cell here connects to the local edge detector, and the fast bipolar cell connects to the so-called brisk transient cell. So in a very real sense, the circuit that you're getting is composed of certain kinds of circuit elements that connect up in stereotyped ways in order to produce the necessary function. Let me take a look at a couple of questions. So Jessica Jiang asks, are the neurons in different specialized regions different in shape and form, or is it only that they're wired to each other differently? They are wired differently, but they can also be different in shape, form, and function. They can use different neurotransmitters, different kinds of things like that. But, and their shape is often adapted to the function that they have to perform. Okay, so let me see. Great, I'm going to move on to the next question. To the next slide. Great, so overall, let me just sort of give an overall summary of what we've said. We've said so far that at every scale of organization, the brain has a diverse repertoire of functional units whose coordinated activity produces the desired overall function. So at the scale of the whole brain, everything you do, you might think about the brain being covered by brain areas. Different areas carry out, vision and audition and olfaction. But then if you look at a smaller thing like retina, which does early vision, well, the kinds of space of things it needs to do get covered by different kinds of cell types, different kinds of computational elements that do the different kinds of algorithms, if you like, that need to be carried out. So a view you might take of this repertoire of computational elements in the brain is that they are a memory of the computations that have predictive value for behavior that have been learned over evolutionary time and then encoded in the genome and in the developmental program. So, so that's a view you could take of how the circuits are organized. So my own lab, my own group focuses broadly on the question of what are the organizational principles or laws, if you will, that control the computational and information processing repertoires of the brain. So, as a physicist, one of the things that's most intimidating when you look at a living system is the extraordinary diversity of forms and functions and all the details that go into that. That's one of the reasons why when I was in high school, I basically, and all the way through college, I avoided taking any courses in biology. I just couldn't take the detail. It was just so complicated. So now that I actually work in biophysics, I think the interesting question for me to think about is to try to bring order to this huge mass of detail. What are the organizational principles or laws that are controlling this diversity and are controlling the organization of these circuits? So that's what I personally try to go after. Okay. So how would you address such a question? So there are many ways and approaches you might take. So today I'll talk about one such approach. So I'd like to think about, well, we already said that the brain is a very limited device. There's a certain number of neurons, there's a certain number of circuits you can build, and you have to do a great deal with it. Now, if you think about the brain in that light, especially as compared to a computer, you'll find that the brain is actually, in some way, an extremely expensive device to have. So there's a constraint here because of that. So the brain, for example, this thing here inside your head is about 2% of your body weight, but fully 20% of your metabolic load. So in other words, it costs you more than muscle when you are working out. Meanwhile, the brain, every millimeter cubed of the brain, so if you put your fingers together like that, it's like a millimeter cubed. It contains 4 kilometers of wire. So this is a very, very heavily packed structure that is extremely expensive to maintain. So that's clearly a constraint, right? It would be circuits and algorithms and structures and circuit elements that wind up costing less or are cheaper should presumably be selected for over time. Indeed, on the other hand, on the one hand, having said how expensive the brain is, it's also true that it's remarkably efficient. So the brain, in the end, consumes maybe 12 to 20 watts of power. So that's like a refrigerator light bulb. And that's much, much less than the laptop that I have here, which is like 80 watts of power. And yet, the laptop can't do poetry appreciation. So one approach that you might take to understanding the architecture of the brain is to say that the brain achieves its efficiency and its efficiency to sort of get over the kind of constraints of metabolic expense and packing circuits and everything like that, that achieves its, that the answer to the circuit repertoire of the brain lies in understanding how to construct efficient circuits. So in other words, you might suppose that the way to understand the huge diversity of circuit elements and how they're organized and so on is to say that by specializing the circuit repertoire and adapting to the structure of the world, you could achieve efficient circuits, and that evolution must be selecting over time for efficient circuits because it's so darn expensive to own a brain. So there's a sense in which this argument is essentially a kind of economic argument, if you like. So, you know, if you talk to sociologists and economists and everything, they'll tell you that back in the day, we were all, you know, hunter-gatherers. We all did everything. You know, everybody hunted the animals and, you know, dug the ground and baked the bread, and you did everything all the time. And society was, you know, maybe not that efficient. And then what happens is that over time, you know, different units in the society, different individuals specialize in different things. The baker breaks the bread, the shoemaker makes the shoes, the hunter hunts the animals, etc. And each becomes more efficient in their task. And together, by exchanging information and goods, this whole society becomes more efficient and more effective. You know, civilization develops, what have you. So in a like manner, you might imagine that the reason why there's such an intimidating diversity of structures, circuits, you know, neuron types and stuff like that in the brain could be that all of these things are organized to specialize in doing different tasks, so that together, by exchanging information, goods, and this, that, and the other, they are able to carry out the tasks that are necessary for animal life within the constraints of how much you can eat, how much power you can spend, and so on and so forth. So we will try to take that approach to try to understand something about the structure of neural circuits. So today, I'm going to try to explore this hypothesis that towards a theory of architecture in the brain by suggesting that brains exploit structure in the world to efficiently allocate limited computational resources to maximize gain for the organism. So we're going to take this idea and run with it and see how much we're able to learn about you know, various kinds of systems. Specifically, we're going to do this by example. We're going to consider 3 examples. We're going to consider vision, the sense of sight, spatial cognition, the sense of place, and inference and model selection. I was tempted to call this sense of truth, in the sense that this is about, you know, can you infer what's truly going on in the world? Anyway, so we'll consider 3 such examples. And in each of these cases, I hope to demonstrate to you that evolution seems to have exploited sophisticated mathematical principles of information processing that have only recently been discovered. And this is supposed to give us a hint that we can use the idea that the circuits should be exploiting, you know, efficient techniques of information processing to build up a theory of organization of circuits in the brain. So that's what we will try to do. Let me take a look at the questions. There's a question. There's an excellent question from Jack Taylor, which says, as the brain adapts to a change in the structure of the world, what is it trying to maximize or minimize? Now, therein lies a complicated question indeed. So I am going to take the point of view that at least in the early visual system, for example, that it's trying to maximize the amount of information you get from the world. And we'll debate whether or not that is an adequate account in a minute. So we're going to try to look at that in a bit. Later on, I'll take the point of view that actually what it's actually trying to do is, you know, it needs, in our animal behavior, requires certain kinds of information, certain amounts of it. So what you should do is you should construct circuits that, let's say, use as few neurons or use as few neural resources in order to carry out that function. So we'll flip back and forth between these different kinds of perspectives. And that's part of the art and interest in trying to do this kind of analysis, identifying what are the constraints that affect neural circuits. What is it that, you know, if you like, evolutionary selection in this context cares about? Whoops. Great. So I think I have answered most of the questions here. Great. Okay. So let's begin. So we'll first begin with the example of vision, the sense of sight. So and I want to discuss one aspect of the circuit repertoire of the retina. So this is and of the cortex. So this is based on a series of papers that are listed here with various talented students and postdocs: Dutch Ratliff, Kasper Tkaczyk, Anhar Munstead, John Briguglio, Tiberiu Tisilianu, and others. Okay, so let's consider the retina, which is kind of the workhorse example that we might have. Maybe the simplest and best understood of neural circuits. So as I already told you, this is the architecture of the retina. Here are the photoreceptors that convert light into electrical signals. Here are the bipolar cells that do some kind of analog processing. And here are the ganglion cells that take the analog signal and produce a digital output, spikes on, they produce voltage spikes that are signaling things to the central brain. And we also said that there are lots of lateral connections here. There are these so-called amacrine cells and horizontal cells that are all inhibitory neurons that remove information coming through this sort of feed-forward stream, but let's leave those alone for now. So the retinal ganglion cells, which are the output cells of the retina, as we said, detect features of the world. There are some that detect bright spots. These are so-called on cells. Here's a bright spot detector. It detects a brighter spot against a darker background. And then there are cells that detect dark spots. These are called off cells. They detect a darker spot against a brighter background. Some detect color, local motion, etc. And they send this information to the central brain. So suppose I ask the question, I'm going to give you a budget, let's say of 1 million cells in the human, 100,000 in the guinea pig. And in order to see well, I could ask, how should the repertoire of ganglion cells, namely these different feature detectors, be divided up into types that respond to these different features? The reason this is a meaningful question is, of course, if you devoted all your cells to, let's say, detecting bright and dark spots, you'd have no color detectors, for example. Right, so there's a trade-off here. Let's suppose there's a constraint that says you can afford, let's say, 1 million cells, then you've got to divide them up into how many do one versus the other, and this gives you some amount of resource with which to do that computation. So I'm going to take an example, and the example I'm going to take is that of bright and dark spot detectors, these so-called on and off cells, and we're going to try to consider, suppose you had a budget to build a certain number of spot detectors, how many should you make? Bright spot detectors versus dark spot detectors. So in all three of the examples I'm going to give, I'm going to start off by doing the theory. I'm going to ask the theoretical question, how should I do X? And we're going to come up with a prediction from that, and we're going to compare the prediction to the experimental data. So that's the plan. Okay, so what should determine the relative proportion of bright and dark spot detectors? Well, on and off cells. So, fundamentally, since in the end, the retina is trying to process natural visual scenes, this must have something to do with the structure of light in the world. So let's start with that. So suppose I consider, what does the world look like? Here is an image from a primate reserve in the Okavango Delta. These are baboons in the Okavango Delta. And this image is taken from some large image database that we collected there. And let's ask about the structure of light of the visual signal in such natural scenes. If you look at such natural scenes, you'll find that in every color channel, red, blue, and green, if you like, you will find that the distribution of light looks like this. It has a low peak and a long tail. It's roughly log normal. The x-axis here is in units of isomerizations per second of the photoreceptors. So you can take this to be some measure of luminance. And if you just count up how many pixels there are, which will generate a certain number of isomerizations per second, you get the sort of log normal type of curve. Now, the thing that's going to be relevant for us is it has a low peak and long tail, which means that the mean exceeds the median. So that's an important point that let's remember. What's more, if you look at natural scenes like that, you'll find that they have long-range correlations. So if you compute the two-point correlation function between 2 points like that, for example, as a function of separation, you'll find that there's a very long correlation length. And indeed, if you Fourier transform this in order to put this whole thing on a log-log plot and put that on a log-log plot, you'll find more or less a straight line. So the phase average power spectrum, namely the Fourier transform of the two-point correlation function of light in visual scenes. So that power spectrum scales as 1 over the spatial frequency squared. So this is on a log-log plot. So if you put it not on a log-log plot, you'd see a 1 over k squared curve. So this kind of scaling of the power spectrum indicates that natural scenes are scale invariant, and that's going to be important for us. What does scale invariance mean? Well, suppose I took the image right there on the screen here and chopped it up into quarters and took 1/4 of it and blew it up to fill up the screen again. You would find that the correlations in that blown up picture are identical to the correlations that were already there. So scale by scale, the structure of the correlations is the same. So let's take away these two facts about the statistics of natural scenes. The first is that the mean light level exceeds the median, and the second is that the correlations are scale invariant. Okay. I'm going to show you from this that, in fact, that there are more, actually, even before we look at this, we can argue from this that there are going to be more dark spots like this than bright spots. So by dark, we mean dark relative to the near surround. And by bright, we mean bright relative to the near surround. So how can we see this? So imagine that this central region, the central region where we're measuring bright or dark, is very small. Then, statistically, the value, the average value of the light here is going to be closer to the median, because it's like, imagine this is 1 pixel. If it's 1 pixel, then, roughly speaking, you can imagine that it sort of falls near the median. On the other hand, the larger surrounding region is averaging over lots of light over a large area, so it's going to be pretty close to the mean light level. So what's going to happen is that the center, the light in the center, minus the light in the surround, if you like, that's a measure of whether you're bright or dark. If that difference is positive, then you have a bright spot. If the difference in the center minus the surround is negative, you have a dark spot. So because the surround is going to be closer to the mean, and because the mean exceeds the median, you're going to find that center minus surround is typically negative. In other words, you'll have more dark spots than bright spots. So just by using these two facts, the mean exceeds the median, by using this fact that the mean exceeds the median, we've concluded that there should be more dark spots than bright spots. Then, suppose we make the scale, if you start with a single central pixel, and then we sort of scale it up, we scale up this on or off cell to make it look at bigger and bigger region of the world, the scale invariance of natural scenes implies that whatever we conclude for very small retinal receptive fields, for responses over a small region, that will scale up. So we can conclude that over many visual scales, it's going to be the case that you get more dark spots than bright spots. So that was just straight reasoning. But now what we can do is we can try to see if we can check this kind of, if you like, experimentally. So here we go. So imagine here's a little piece of a natural scene, and I'm going to take on cells and off cells like this, and I'm going to take a little difference of Gaussian's model where I take a central Gaussian minus a surrounding Gaussian that measures the light response, and I'll normalize it, let's say, by the surrounding Gaussian. So I'm going to filter the scene with a central Gaussian minus a surrounding Gaussian, and then normalize by the surround. This basically measures percent contrast. And you can measure at each point whether you get a bright spot or dark spot, right? So here we've isolated all the on responses, places where an on cell would respond. Here we've isolated all the places where an off cell would respond. And then if you go through the entire scene, you discover that no matter how big you make the visual angle, that's the center sigma is the standard deviation of the central Gaussian. No matter how big you make the visual angle at which you're looking at things, you'll find that there are more dark spots than bright spots. In other words, that confirms what we sort of argue should happen through pure reasoning. The fact that you have the mean light level at pixels exceeds the median light level causes there to be more dark spots than bright spots, and the scale invariant correlations cause this effect to persist across scales, no matter what visual angle you are looking at. Okay, there are some questions. There are a whole bunch of questions. Actually, I'm not sure whether I should be going down in the question list or up in the question list. You know what? Perhaps I'll take these questions at the end of this section. Okay, so we'll come back to this in a bit. So great. So now, given these statistical facts about the nature of light in natural visual scenes, we could ask how to best design a detector array. So we're going to assume that you're bequeathed with a constraint which says that in your on-off channel, namely in the part of the visual system that's going to process bright and dark spots, you are able to afford n cells. Let's suppose for whatever reason there's a constraint from the amount of power you can expend, or there's a constraint from something else that says that you can only have n cells. So you can ask, given that you need to split these n cells into a certain number of off cells, let's call that n off, and a certain number of on cells, let's call that n on, we can say, how should we find the off to on ratio that maximizes the total information? So the idea is, You have a certain number of cells and you want to see well, so I want to maximize visual information, and I have to divide the number of off and on dark and bright spot detectors in such a way that maximizes total information. So how can I do this? Well, suppose your budget had been that you could only have one cell. Then the answer is clear. You should choose an off cell, a dark spot detector, because it's more likely to respond. Done. So then as you increase the number of cells, what would happen? Well, so initially you'll sort of cover the retina with off cells, dark spot detectors, but then pretty soon they're going to start overlapping and get redundant. So if they get redundant in their responses, obviously it's going to be beneficial to put in on cells. So then you'll start putting in on cells, bright spot detectors, and pretty soon they've covered the retina and they're getting redundant. So then you should put in an off cell, and then you should put in an on cell, and you'll go back and forth like this. And it's clear that this game we're playing to design the, if you like, the best detector array, the best retina, if you like, that this game we're playing can only terminate at some balance point where the individual off and on cells convey equal amounts of information. Because otherwise, if they didn't convey equal amounts of information, it's beneficial to add the other type. So that's going to have to be the answer, that whatever theory, mathematical theory we construct should give that as the answer. So indeed, so let's construct now a very, very simple theory. We're going to say that the total amount of information in the array is the information individual on cells times the number of on cells times some factor that controls for the redundancy in overlapping on cells. Likewise, the amount of information in off cells, in the off responses, is the amount of information conveyed by individual off cells times the some factor controlling for the redundancy of off cells times the number of off cells, and the number of off cells is the total number of cells minus the number of on cells. Now, in information theory, there are many ways to talk about the amount of information conveyed by system. And the simplest, since we're here building a very simplistic theory, is to say that, well, you know, suppose I work out the signal to noise ratio of a cone. That's a measurable quantity experimentally. And then I say that each retinal ganglion cell is looking at a piece of the world. And as it looks at a piece of the world, it sums over some part, some number of retinal ganglion cells with some, you know, weighting factors. So this, so suppose it does that. So you can imagine that the retinal ganglion cell output, the output of the retina, is some weighted sum of its inputs. So if you do that, you can work out a factor here that tells you how the signal to noise ratio of the cone is improved by summing over many cones. And that's this factor f squared on, which I'm not going to calculate in detail. And then in a very simple Gaussian approximation, the amount of information conveyed by, let's say, the on cell is 1/2 the log of 1 plus the signal to noise ratio of the cones, enhanced by the sort of summation over neighboring cones to beat down the noise. And it's a simple exercise, if you do this, to work out that if you now optimize this expression for the information, namely you maximize with respect to the number of on cells, you're going to discover that at the optimum, the individual on cell has the same amount of information as the individual off cell, as we argued already, if you like, from pure logic. Okay, so of course, this was a very simplistic theory. You can make this more sophisticated, and we're not going to go through the details of that. That was the content of one of the 3 papers that I asked you guys to read for today's session. That's the paper Ratliff et al. From PNAS in 2010. So you do some calculation in a bit more detail, a bit more faithful to the actual physiology of the neurons, and you can work out that in a realistic model, the optimal ratio that you should have is you should have 1.7 times as many off cells as on cells, roughly. So there's some error bar there. So, okay, so then there's a prediction here from the theory that if this kind of idea is true, that the retinal circuit is selected to maximize the amount of information conveyed from the world, then in the on-off channel, namely bright and dark spot detector channel, you should have, you know, somewhere between 1.5 and 2 times as many dark spot detectors off cells as on cells. So what does the data say? Well, so first of all, there is now substantial evidence that the brain separates light from dark unequally. So behavioral measurements have shown greater sensitivity to light decrements and dark spots in images. Likewise, there are experiments that show that there are more cortical cells that respond to dark spots, namely negative contrasts, than to bright spots, namely positive contrasts. And then the key point is about the retina. And across a variety of species, guinea pig, rabbit, rat, monkey, human, cat, there's a considerable both anatomical and physiological evidence that exactly as predicted by the theory, retinal off cells are somewhere between 1.3 to 2 times as numerous as on cells. And this is predicted because there are more dark regions in natural scenes and because information is more densely packed in them. That's pretty cool. You know, that was a highly non-trivial thing about the structure of the retinal circuit that we've been able to predict just by thinking about how you encode visual scenes efficiently. So you could ask the question whether such an approach could be applied generally. So going back to one of the questions that was asked earlier, well, what's being optimized? Well, so here I took information as an approximation of the objective function of the early visual system. Well, so that's just an approximation. And in other situations, it may not quite be the information per se, but there may be some value associated with the information, et cetera. So there, you really have to understand what it is that the circuit or the animal cares about, and then you should ask that that gets optimized. Another question that you might ask, and that's often, this is a question that a biologist would ask, well, why are you sort of optimizing this, that, and the other? Shouldn't the forms and functions just be determined by the evolutionary history? One way of thinking about this is that, you know, personally, I would think that I would be more optimal if I had a long tail with an eye at the end, so that I could look in high places or something. But I'm never going to get it because I'm the wrong lineage. I don't have such a thing within my evolutionary lineage. So here I would say that really the way there's a subtlety in applying these kinds of optimization principles. You've got to think within an evolutionary lineage, which is constrained by its history, you might say the better adapted forms and functions are selected over time. This is a little bit like saying that, you know, greyhounds and cheetahs run fast and therefore they have a similar body form because, you know, whatever the lineage is, they have got to do something. And so within that lineage, their body forms adapt in such a manner that they can run fast. So within the lineage, you want to adapt in such a manner as to do the task well. Another good question is, well, I'm optimizing things in the analysis that I showed you, but in real life, nothing is ever optimal. So life is effectively a work in progress. So why should anything be optimal? So I think the correct answer to this is somehow, really, it isn't, right? Nothing is ever optimal per se. So, but really, we should be thinking about the optimal solution as a guide to the principles underlying circuit organization. You know, if I had a way of mathematically formulating the notion of pretty good or very good, I would. But I don't know how to formulate that. I can formulate optimal. So I'll formulate optimal and take the prediction of the optimization theory with a little bit of a grain of salt. It's a guide to an operating region where you're predicting the circuit will live. So another way of thinking about why it's worth doing this is it's an approach to answering the question of, is there some order in the sort of absolute chaos and diversity of the structure of neural circuitry? And I once read a book on particle physics which had a foreword by Sheldon Glashow, who won the Nobel Prize for his work on the standard model. This was a book by Howard Georgi on Lie algebras and particle physics. Okay, so in this foreword, Sheldon Glashow says something like, there may not be any order in nature, but those of us who look for it have a better chance of finding it if it is there. So I think these kinds of approaches to understanding the structure of neural circuits should be taken in that light. It's possible that there is no real organization there and that, you know, it's just some crazy evolutionary mess. It's just the history of the organism and things like that. But if it's there, if you look for it, you're more likely to find it. And that, I think, is the job of physicists like us. Okay, so a way of trying to answer this question, can this approach be applied more generally, or does it apply just very, very specifically to, you know, the early visual system or something like that, is of course to look for more examples. So well, already in vision, there's now considerable evidence that these kinds of approaches can be applied more generally and can be, for example, extended to thinking about the cortical vision. So we don't have time to go about discuss this in great detail. But for example, so I'm going to give this to you very schematically, and then we're going to move on to the second example of cognitive processing. So imagine, instead of thinking about the detection of things like spots and color and stuff like that, we think about something that's properly cortical. So for example, your ability to perceive texture is essentially a cortical, involves cortical processing. So by texture, I mean, you know, suppose I take an image like this on the left and I look at the correlations between the bright and dark pixels at different points. You know, the pattern of correlations between the bright and dark pixels at different points is what defines a texture. A visual texture is defined by that. And you can do experiments where what you do is you construct various kinds of artificial textures, which have certain patterns of correlation. Look at these, for example. All of these are different patterns of correlations that produce, amongst pixels, that produce visual textures. And then you can do behavioral tests, psychophysical tests, like you take a background of white noise and you put a little strip of one of these structured textures on that background, and you give somebody 150 100 milliseconds to tell you whether or not the whole thing is white noise or whether there's a little bit of texture of structured texture in it. So you can do psychologists and behavioral scientists have done many tests like this. So what can people detect? So if you follow the same kind of logic of the theory we just presented for the retina, you should say, well, your ability to detect texture should be related to the usefulness or the informativeness of specific textures about the natural visual world. So one way of extending the kinds of ideas I just talked about is to go and look at the distribution of textures in the natural world, and then ask if you want to maximize the amount of information you have about the texture content of the natural world, how would you devote your neural resources to detect this kind of correlation or texture versus that kind of correlation? And you'd predict that those textures that vary more across the natural visual world are more useful to you, clearly, in detecting, are you looking at this, are you looking at that? And so that therefore you will somehow devote more neural resources to detecting textures that actually vary a lot in the natural world, and thereby your threshold for detecting those correlations will be lower because you'll be more sensitive to them. You'll spend more effort on them. And indeed, again, we're not going through this in detail. In these papers, we have shown that there is very broadly a principle that you might call variance as salience, that textures that vary more between image patches in the natural world are more informative about images, and thus more neural resources seem to be devoted to detecting them, and hence your detection threshold is lower. It's easier to detect those textures that vary more in the natural world. And that follows very naturally from the kinds of information theoretic principles that I was discussing a few minutes ago. I'm just putting these papers up here because if you're interested, this is one place where you can follow up and read more about it. And there are also lots of references in these papers from which you can learn more about this kind of approach to thinking about sensory systems. Okay, let me see if there are any questions that I should answer. Okay, so Abdul has a question which says, is this maximization, the kind of thing I'm talking about, specific to more highly developed brains, for example, mammals, or general to all organism brains? So these kinds of ideas about the organization of neural circuits have been applied all over, and even to things like C. elegans, which basically has a network of a few hundred neurons, and there isn't a central brain in the conventional sense per se. They've even been applied to single neurons, like single neurons detecting signals. For example, there's a famous paper by Simon Laughlin talking about the response properties of the so-called large monopolar cell in the fly, which detects contrast. It's like an on and off cell, and it describes the on and off the properties of this LMC cell by using similar ideas of information optimization. Okay, so great. So I'm still looking through the questions. I think I've answered a couple of them. So I am going to move on. So, oh yeah, so there's another question here that says, has there been work that looks at quantities for the partial information decomposition of this mutual information? I would think that optimizing the synergy or the complementary information between the on and off cells is worthwhile. So the answer to that question is absolutely, that's true. And in fact, the full calculation of the optimal proportions of on and off cells involves trying to pay attention to that kind of synergy, because these are two complementary channels and they interact in some way. Great. So then I'm going to move on to the second example. So, but before that, I'll set a challenge to the students here. So there are quite a few of you logged in. So here's my challenge. So in the talk today, which referred to a paper of mine from 2010, we discussed explaining the relative proportions of on and off ganglion cells by talking about how much information they conveyed about the visual world. Well, you know, there are about 20 different kinds of retinal ganglion cells. So here's my challenge to you. Use similar ideas or other ideas to explain the relative proportions of all of them. That way, if you do that, you have given a complete theory of the design of the output features of the retina. So that's a challenge for, let's say, the next generation. Okay, I'm now going to go to my second example. So here we're going to talk about a repertoire of circuits that deals with cognitive function, namely the sense of place. So this is a much more subtle thing because you see sensory systems, you know, deal with the real visual or auditory or olfactory worlds. So you have a lot of control over the signal that the circuit is trying to process. But when you talk about a cognitive repertoire, there's something going on deep inside the head. So I'm going to talk about the sense of place. So first of all, we should ask, what is place in the first place? How do you know where, how do you even know where you are? So inside your head here, a location is some sort of abstract pattern of neurons firing. And these sorts of patterns maintain a map of your physical location. So like we did with the on and off cells, I want to start off thinking as a theorist without looking at the data. And as a theorist, I might say, well, okay, so suppose I want to make a map of space. What would I do? So here we imagine a simple one-dimensional world. And in this one-dimensional world, let me suppose there's a 8-meter track. And for my behavior, let's suppose that I need to know where I am with 1 meter resolution. So one way of constructing a neural representation of this world is to imagine that you have, we can call them place neurons, each of which fires if the animal lives in one of these 1 meter blocks. So you have 8 blocks there, so you need 8 neurons to represent this location. Okay, so that's one way of doing it. Here's a more efficient way of doing it. I imagine that I have one neuron, this blue neuron, that responds in the left half of the room, and this orange neuron responds in the right half of the room. Then I have a second scale where I have the blue neuron responding in the left half of the room and in the left half of the right half of the room. And the orange neuron responds to the right half of the left half of the room and in the right half of the right half of the room as shown here. And you can do this recursively. So if you do that, if blue is 0 and orange is 1, this location here is 0, 1, 0, and this location here is 1, 0, 1. In other words, this is basically binary code for space. And notice that it requires only 6 neurons. In other words, it's slightly more efficient than the place scheme, which required 8 neurons. And you can use other bases like decimal. But when you walk around the world, typically you're doing that in 2 dimensions. So here, let's talk about representations of a 2-dimensional space. So once again, let me imagine the world is 8 meters by 8 meters, and that you need a resolution of 1 meter by 1 meter boxes. So well, again, you can have a place scheme inside your head to represent this, where you have neurons that respond, a neuron that responds when you're in any one of these locations. So if so, there are 64 such locations, so you need 64 neurons that can tell you where you are. So the firing of a particular neuron tells you where you are in this world. Now, let's do a 2-dimensional analog of binary numbers. So 2-dimensional analog of binary numbers might work like this, right? You divide the room into 4 blocks, blue, orange, yellow, and green. And the cross represents where the animal is. Now I recursively subdivide the blocks again. I divide this blue block into 4 parts. Then I take any one of these little blocks, the smaller blocks, and divide that into 4 parts and so on. So if you do that, the idea here is at the largest scale, the blue neuron responds in the top left corner of the room. At the next scale, I might imagine a neuron that responds here and here and here and here. In other words, in a lattice of locations that are in the top left corners of each of these sub-blocks, and so on. So on recursively. So basically, we imagine neurons that are responding on a lattice of locations. So if the animal is anywhere, let's say on the blue lattice, the blue neuron responds. If the animal is anywhere on the yellow lattice, then the yellow neuron responds at this scale. And one scale down, there is a yellow neuron that responds at all of these lattice locations, etc. It's a very Baroque scheme, you might think. On the other hand, notice that this is vastly more efficient than the place scheme for encoding 2-dimensional location. So in particular here, you need only 12 different kinds of neurons to locate, to tell you where you are. So for example, this location is the blue in the top scale, yellow in the middle scale, and blue in the smallest scale. And so with 12 neurons, much less than the 64, you can encode space completely. And you can use other kinds of bases too, like decimal. So you can use, for example, base 10. So now I'm going to try to argue to you that the brain actually has a system like this. So there are, in a region of the brain called the Internal Cortex, there are cells that respond when an animal is physically in locations lying on a triangular lattice. So for example, if this square box is the room, the hotspots tell you where the animal is when this neuron responds. So it turns out that if you look at the neighboring neuron, it'll vary randomly in offset or in phase. And then as you go deeper along the axis of the entorhinal cortex, it turns out that the grids increase in scale. So there's a bigger scale and a bigger scale. And what's more, they're organized in discrete modules. So it'll turn out that there's this period, and if you go deeper in, the period jumps. And then if you go deeper in, the period jumps by a different amount. And this is work by the Moser lab that revealed these things. So I'd like to draw your attention to the analogy between this kind of structure where you have neurons that respond when the animal is in a lattice of location, small location, bigger location, biggest location. I'd like to draw the analogy between that and the picture we just drew of a binary-like code, right? So in other words, the grid cells remind one of some sort of two-dimensional fuzzy number system for location in a two-dimensional world. So you could ask the question, well, how would I justify that that's how I should interpret the grid system? Well, one way to justify that is, I've been trying to argue that efficiency is a principle that governs the structure of neural circuits. So, well, we already said that a unary system, namely a place system, required 64 cells in the binary, to encode, in the example that I gave, required 64 cells, and a binary code required 12 cells. So I could ask, which number system should the brain pick? And how about picking the most efficient number system that requires the minimum number of cells to encode space? So we can try to make the prediction for what number system the brain should be using. Now, I should say that there's an important subtlety. When we write down numbers on a piece of paper, the base of the number system has to be basically an integer, right? You have base 2, base 3, base 4, because we've got to write down the numbers. But the brain doesn't have to write down any numbers. So just like base 2 involves subdividing space by factors of 2, and base 10 would involve subdividing space in factors of 10 recursively. In the brain, if you look at these grid cells, the base of the number system is in some sense the ratio of the periods between adjacent modules. So if you take the ratio of this period and this period, and this period and this period, that ratio is like the base of the number system for location. So we're going to think about that and try to make a prediction for what base the brain should be using the same kind of efficiency arguments that we were trying to use in the retina. This is, of course, a much harder problem because we're deep inside the brain and we don't have access to the simple visual inputs or anything like that to the structure. So how are we going to do this? So here, it's easiest to first think about this in one dimension. And then after thinking in one dimension, we'll think in multiple dimensions. So imagine here is a grid system. Here at this first scale, the red cell responds in this region, and the blue cell responds in this region. At the next scale, the red cell responds here, and the blue cell responds here and here, and so on. In other words, it's a lattice of locations at which physical locations for the animal where the cell responds. Now, suppose here's another grid system where you might think that you can reduce the number of cells you need by sort of shrinking these grids as fast as possible so that you get better resolution, right? If these response regions are small and compact, you might think that you could get better precision in your location. But look what happens if you try to squish this in too quickly. So suppose you take this top scale, and then at the second scale, you squish in a lot so that the spacing, so that the period at the second scale is smaller than the width of this response region, the red region in the first scale, then you're going to get an ambiguity in the encoding of location. What I mean by that is the following. So suppose in this first scheme, the red cell responded at the top scale and the second scale. Then you knew for sure that the animal was here because the red cell responded, and that furthermore it was here because, well, this is a subset of this. But in the second scheme here, if the red cell responds at the upper scale and then the red cell responds at the second scale. You don't know if the animal is here or here. Those are both consistent with the red cell responding at the upper scale and the red cell responding at the lower scale. So in other words, in order to avoid ambiguities, you're going to need the period of the grid field of the grid system of the grid cell at the second of the smaller scale to be bigger than the width of the response regions at the upper scale. Okay, and you can do this in a more real neurons don't respond evenly within little square grids like this. Really, they have sort of more smooth response fields and everything. So all of this needs to be sort of modified and improved or be corrected to account for the real response properties of neurons. We're mostly going to stick to a schematic of this kind, but all of the calculations can be generalized to a more realistic setting. Okay, so then what do you do to optimize things? Well, again, although I'm drawing two-dimensional pictures, I'm actually doing the calculation here for one-dimensional grids to keep things simple. And later on, I'll tell you what the answer is for grids in any dimension. So what are the parameters of such a system? Well, you have narrow grids at the dorsal end of the entorhinal cortex, and you've got big grids at the ventral end of the entorhinal cortex. Each grid is characterized by its period, here labeled lambda I, and the width of the region where it responds, here called Li. So you can parameterize this sort of scaling hierarchy by the ratios of the scales. So you take lambda I over lambda I plus 1, and then you have lambda I plus 1 over lambda I plus 2, and so on. Those are these ratios of grid scales. Now, what's the resolution of the grid? The resolution, which is what you want to fix, is the fineness with which you can chop up the entire space. And that's going to be the largest period divided by the smallest period. That roughly tells you the number of pieces that the grid system can break up the room into. How fine is the map you can create of the world? Now you can see that this is just simply a that lambda, the biggest scale over the smallest scale, can be written as a telescoping product of the ratios of grid scales. That's the first parameter we have. A second parameter has to do with the number of grid cells. So you see, in order to cover the world, you're going to have grids of many different phases. So if you have this grid cell localized here, you can need one that's shifted there, another one that's shifted there, and another one shifted there, etc. So basically, what's going to have to happen is that at each scale, the number of grid cells you have is going to have to be proportional in one dimension to the period divided by the width of these things in order to cover the world. Okay, so that's a second number that we have. And there's a there's a bigger period than a smaller period, and so on. And so suppose we want to minimize the number of resources you need to use in order to describe the world. In that case, what you're going to have to do is you're going to have to minimize the number of cells for a fixed resolution R. And okay, so suppose you want to do that, you have a constraint. We already said the period at next scale, I plus 1 scale, must be bigger than the width of these firing fields at the ith scale. And you can translate that into some kind of constraint on the periods and the widths of these firing fields. So if you put this all together and sort of grind through the maximization, it's a very easy problem to do. And you can show that you make a bunch of predictions. The predictions are the ratios of adjacent periods will be equal. That is to say, all of these RIs are equal. And that this ratio, to optimize or minimize the number of cells required to represent space, should go as the dth root of E, as it turns out, in D dimensions. So in 2 dimensions, that's the square root of E. And there's some additional predictions. So we can compare this with the data, right? So if this theory is correct, then this prediction for the ratio of grid scales should be correct in the brain. So if somebody goes and measures the period at the dorsal end and the next module, the next module, the next module, and computes these ratios, the prediction would be in 2 dimensions. They should find that those ratios, amazingly, would go like the square root of E. So strictly speaking, to really apply this to the data, you have to be a little bit more careful because neurons are noisy. They respond, with some probability in different locations, So you have to do similar kinds of things about resolution versus ambiguity with more probabilistic decoding. I'm not going to go into the details of that. You can go check the details in the papers. But what you find at the end of the day is that the theory makes a prediction that the simple model makes a prediction that the scaling ratio of grid scales should be lie in this range. And the fully probabilistic model that I told you about, the more sophisticated model, makes this prediction. And here are 2 data sets. This was the first data set reporting these kinds of ratios. And then this is a massive data set with hundreds and hundreds of cells. And it's a beautiful match with the theory. So evolution seems to have invented here base n number systems and then optimized them for neural hardware. So this seems to suggest that an efficiency principle seems to explain the organization of complex circuits supporting a cognitive function. So that, I think, is pretty amazing. And this is a wonderful confirmation of the idea that we can use this kind of approach to try to understand the structure of neural circuits. Now, the lecture has already run over, and I had two more things that I could have discussed. So I'm going to leave one of them for the question time. So the one question you could immediately ask is, suppose you have this sort of complicated structure involving scaling hierarchy of grids. How do you build that? How would the brain build that? Can it dynamically self-organize it? Can it, or does this have to be hardwired in? So we have done a bunch of work using basically techniques from dynamical systems theory, showing that you can self-organize such a structure. So basically, as it turns out, these kinds of grids in the responses of neurons in the brain are learned as an animal explores a new environment. So that suggests that the grids provide learned and self-organized cognitive map. So really the question we have to answer is, there's supposed to be this amazing scaling hierarchy with this sort of, there's a grid, and there's another grid which is sort of 1.4 to 1.5 times as big. And there's another one that's even bigger by 1.4 to 1.5, and that this is what the brain uses to create and keep a map of the world, and that we have to be able to learn this map then every time you come into a new environment. So, so you need to have a self-organizing mechanism, and indeed in this paper we describe such a self-organizing mechanism. At the end, I want to, if people ask me questions at the end of this talk, I can return to this and discuss this self-organization mechanism. But for the moment, I think I am going to actually move on to discuss the third example, which I think is quite exciting and is related to some of the things that Eugenio Piacini discussed in the discussion session earlier today. So far we've seen two examples of the use of these kinds of efficiency ideas to discuss the structure of circuits in the brain. One was in the visual system, where we talked about the relative proportion of dark and bright spot detectors. And the second was in a cognitive circuit. This is a circuit that is used to maintain your map of physical location in the world, right? Now I'm going to think about something even more abstract, which is how the brain selects models of the world. So this is, what do I mean by this? Well, you know, animals live in an uncertain world, right? And somehow in order to know what to do next, you need to know something about, even if you want to use your map inside your head, and suppose you want to use your visual input and the map inside your head in order to decide what to do next. Well, you need to know what the world is and what the world is going to be. So this requires all animals to have ways of knowing the latent states of the world and then predicting what the future is going to be like to enable planning and action, right? The problem is, of course, the current and future states of the world are noisy and the cues are uncertain. That is why this person to the left here looks confused, right? And what's more, you know, the world is changing constantly and we have to keep updating our models. So how are we supposed to do this? So again, I'm going to take the attitude of theorists to say, I'm going to ask how to do this best. You know, what's the optimal way of doing this? And then I'm going to ask, is there any evidence that animals or humans follow these kinds of principles that arise by thinking about the fundamental statistical and computational problem? So, okay, so how are you supposed to do this? Well, you know, in science, we often use this idea of Occam's razor, where suppose I have a bunch of data and I've got to ask, what is the latent process that produces this data? Because, you know, I want to predict when in this figure, for example, if X is, you know, over there, twice as big, I would like to know what the value of Y is. That's like, I see a ball, I see a ball, I see a ball, and now I want to catch the ball, so I need to be able to predict a little while later, that's a little time later, where the ball will be. And I need to be able to make such predictions. How would we make such predictions? Well, you see, if you have this bunch of data, these dark dots, there is a curve that fits it perfectly if you take a degree n-1 polynomial, but it may be or may not be that that's the best way to extrapolate into the larger values of X. It may be that a more parsimonious model, like this linear model, somehow gives a better description if this data was unreliable. So this idea that sometimes simplicity is or less complex model give better predictions goes back a long way to William of Ockham, who wrote that causes should not be multiplied beyond necessity. He was actually talking about at that time the attributes of God and trying to encourage people not to add more attributes to God to explain the world. But anyway, the bottom line is that there's some trade-off between the accuracy of predictions and the complexity of the model you use to match existing data. And there are many ways people have thought about doing this in the past. There's the AIC model, in which you take the minus log likelihood of the data and add to it the number of parameters, then you pick the thing that minimizes this quantity. There's the BIC model, which sort of takes minus the log likelihood, maximum likelihood of the data, and then half the number of parameters times log n. Okay, so we can ask, what's the best way to do probabilistic model selection? So I know Eugenio Piacini in the companion lecture has discussed this in detail. So I'm not going to go over that material again, but I'm going to remind you the way you're supposed to do this is that I have a bunch of data, a data set, n data points, and there's an animal observing the world. And let's suppose the animal has to decide if this data was generated by distribution of the family A or in the family B. Then as Eugenio described to you, the Bayesian approach to doing this would say that the probability of the model family A given the data is the likelihood probability of E given A multiplied by the ratio of priors. And then if you take the ratios of priors to be equal, you can select whichever model maximizes the likelihood of the data. And furthermore, the likelihood of the data given the model family can be calculated as an integral over the parameter space of the prior probability of the parameters times the probability of the data given the particular parameters, right? And I think Eugenio also described to you the choice of prior. If you know something in advance that the parameters of this model should be localized, say here, then you should pick that prior. But otherwise, there's such a thing as an uninformative prior that weights all the distributions in the model family equally, and that's given by the square root of the determinant of the Fisher information, and the Fisher information is this quantity, which is the second derivative matrix of the likelihood of the data given the models. So again, as described in some work of mine from 1997, and as Eugenio would have described in the discussion earlier, there is an approximation you can make of this likelihood function, which looks like this. It says that the log likelihood of the data given the model, and hence the likelihood of the model given the data, given equal priors, is equal to the minus the log maximum likelihood of the data. Namely, you pick the maximum likelihood parameter, and then you evaluate the probability of the data given that, plus half the number of parameters times log the number of data points, plus a quantity that involves the integral of the square root of the Fisher information over the entire model, plus this log ratio, which is the log, the determinant of the empirical Fisher information. Empirical Fisher information tells you about how much changing the parameters a little bit changes the quality of your description of the data, divided by the determinant here of the actual Fisher information in the model, which tells you if you change the parameters a little bit, how much the models around it in the model family vary. So these various quantities, so the first quantity here rewards a model for the quality of the description of the data. So if you get a large log likelihood, it means the model describes the data well. The second quantity here punishes the model for having too many dimensions. So this is like your classic penalty for having too many parameters. The third quantity here penalizes the model for having an overlarge volume. So what this basically penalizes, an over-expressive model isn't very specific. And so because it isn't specific, it's going to describe lots of things well, right? So this is like this situation where this red curve would have a large volume because it's going all over the place, it's not very constrained, and so it should be penalized. It's less simple, it's more complex. And then the fourth term here is penalizes models that are not robust. So let me explain that. So suppose this is the truth T, and here are two models A and B. And let's suppose model B comes closer to the truth at one location, but if you fiddle the parameters a little bit, it rapidly gets bad. By contrast, let's suppose model A doesn't come that close anywhere, but no matter what parameters you pick, it'll be pretty good. So A is more robust than B in its description of the data, and this quantity here rewards the model for being more robust. So what this says is that probability theory naturally produces a measure of the quality of the model, this quantity here, which punishes models for complexity. So this is a very natural sense in which probability theory knows Occam's razor. So what I would like to talk about is whether or not it's the case that people actually follow this principle, right? Does the brain, do the circuits in the brain effectively implement a decision-making circuit, an inference circuit that prefers simpler explanations when performing model selection? And how, therefore, do animals or humans trade off simplicity and goodness of fit? So I'm going to end today by describing data on that. So here's the way we're going to test this kind of inference bias. We're going to construct an experiment where there are two flower beds, let's say the upper one and the lower one. And the person gets told that a flower may be present anywhere on the upper bed or on the lower bed, and that drops 10 seeds. These are the red dots. And the seeds are distributed in a Gaussian with some fixed variance. The flower can be anywhere. So here's the question. Which flower bed has the flower? So in this case, you see this model of the world has a larger volume, and this model of the world has a smaller volume, right? So in this way, you can construct different flower beds with different shapes that test different pieces of this expression. So you get this part, this is the dimensions, this is the volumes, this is the robustness, and by constructing flower beds that are shaped in different ways, you can construct a sort of toy scenario where you can test the influence of each of these different kinds of terms. So indeed, we construct 4 types of trials. In this case, this is a 0-dimensional flower bed and a 1-dimensional flower bed. This is a flower bed which has a sort of boundary, and this one doesn't. This tip is what I'm talking about. This flower bed has a large volume, and this has a small one. And in this case, you'll see that if you evaluate the robustness term, this gives less robust descriptions of data because the tip will come close to the actual data we're going to look at, whereas and then it'll go further away. So this is less robust. It will give less robust descriptions of the data than this one. And so we basically conducted an experiment where 50 subjects were assigned to each task type. They went through a tutorial, and they got many, many chances to try to guess where the flower was. And we tried to ask, do people have a simplicity bias in picking their models? So I'm going to skip to here. And so we haven't discussed in detail exactly how the simplicity bias is measured. But I'm going to explain that to you now. So if you did so it turns out that there's some quantification of the simplicity bias. And here are the 4 tasks. This is the one which tests the robustness, whether you have a bias towards robustness. This is the task that tests whether you have a bias towards models with smaller volume. This is the task that tests whether you have a bias to lower dimensional models. And this is the task that tests if you have a bias against models with boundaries. Okay. So if you just using probability theory, given the data, the maximum likelihood model, right, the most likely answer every time you have the data, then the simplicity bias would be 0. So the red lines here indicate if people use maximum likelihood model selection, then the results from them would have lain on this red line. Now, suppose you use optimal model selection, right? Optimal Bayesian model selection with a simplicity bias. Then we predict that a person's that the simplicity bias would have lain on this green line here, this green line, this green line, this green line, this green line. And the actual people in these tests so far, they are distributed according to these colored curves that you see. So you see in all cases, there's a bias away from the maximum likelihood choice towards what would be the simpler model. In some cases, the bias is almost exactly what would be predicted by the optimal theory. In some cases, it's more. In the case of the dimension, people are more biased towards having lower dimensional models than warranted by optimal probability theory. But in all cases, there is a bias towards simple models. So what does this tell us? It tells us that probability theory has first I told you the theory told us that probability theory has a built-in Occam's razor that should cause simpler models to be preferred until there is enough data. But sort of amazingly to me, humans seem to show precisely such a systematic bias towards simpler models and away from the maximum likelihood choice, thus reflecting the Bayesian prediction. So then that raises the question of how do the actual neural circuit architectures implement this behavioral outcome? Because it must be the decision-making circuits in your prefrontal cortex essentially are discounting for the kinds of complexity penalties that probability theory would tell you to discount for. Okay, so that was the third thing I wanted to talk about as an example. So what I've talked about 3 things today, and really the whole talk was supposed to be suggesting that it is possible for us as physicists to make progress towards a theory of the functional maps and computational repertoires in the brain and how they self-organize, right? So that we can, in principle, that there's some hope to come up with a theory of how all these algorithms are structured in the brain. And today I gave 3 examples, one from vision, one from spatial cognition, and one thinking about statistical inference. In all cases, adaptation to the environment and to the task, the constraints of neural computation, and efficiency and parsimony were the 3 principles that allowed us to think about what is the best circuit, and then, or what is the best way to perform this task. And then we were able to test that the actual circuits in the brain or human behavior reflected those kinds of principles. Animals appear to be implementing circuits and doing things that are well adapted to the environment, that given the constraints of neural computation, and that embody principles of efficiency and parsimony. So all of this is kind of an attempt to explain the sort of diversity and complexity of computational architecture in the brain. That's really the main goal of the work we do in my lab. And I invite all of you to try and think about such things, because I think that we as physicists can definitely make progress on it. Okay, I'm going to stop there. And I'm going to look at some of the questions. Okay, so okay. So I'm going to take questions in some order here. So there's a question from Stuart Reinhardt that I think applies to some of the things that were said about grid cells. And he says, could the logic that I was describing apply for 3 dimensions, for grids in 3 dimensions? So the one animal where we are aware of 3-dimensional grid cells forming is in the bat. There's some data from a lab at the Weizmann Institute of Nahum Ulanovsky, where they have data from 3-dimensional grids. So indeed, this sort of logic in principle applies to that, except there's a question of whether you can self-organize a 3-dimensional grid of that kind. So we didn't discuss the self-organization part of the talk. And so there's a question about self-organization. So that discussion is ongoing as to whether or not we will see that. Let me see if there are any other grid cell questions. So I don't see any other grid cell questions. Okay, so then there's a question from Yujia Zhang, which says, when a large volume is punished, is that equivalent to squeezing neurons together to increase the resolution? So I'm not precisely sure if you mean by this that physical squeezing of the neurons. So that's not what was meant here. So this was meant in a very abstract sense. So the idea here is that the implicit model maintained either in the brain or explicitly, the scientist's computer, if you like, can have a larger or smaller model volume. What do we mean by the volume? So let's suppose a model describes some probability distribution. Then as you vary the parameters of the model, you are effectively doing this. As you vary the parameters of the model, let's say this, you trace out, let's say this red surface, which is some model, which is some manifold. It's a surface in the space of probability distributions. So in some invariant sense, we can talk about the model of volume. So as we said, there's a notion of volume on these kinds of probability distributions given by the square root of the determinant of the Fisher information as the local measure that you have to integrate. And so in that sense, models that have a large volume are less constrained. They describe more possible worlds. So I was talking that abstract language. Exactly how that's realized at the level of neurons or neural circuits that describe larger volume or smaller volume models, that remains to be understood. Hope I answered that question. Otherwise, please put something into the chat box. Actually, there's some kind of answer live box here. Does that mean I can just, what does that mean? Can I click the answer live somehow here? And
yes, when you click in on answer live, the question is going to be dismissed. It will be answered question.
Oh, okay. So I am clicking answer live for that one. I'm clicking answer live for Stuart Reinhardt. Okay. So there's another question here which says, do you think um, um, that machine learning, artificial intelligence training of pattern recognition, oops, what happened to the question? It disappeared. um, do you think that the machine learning, artificial intelligence training of pattern recognition can have a correlation with the natural biological system of pattern recognition? So this is by Duti Das. And I interpret this question as asking, so suppose, say, for example, you construct a deep network to perform such task. Can you, uh, 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 can it be that on the one hand, the structures that you learn resemble the structures of neural architectures to solve a similar problem? And can you maybe use, therefore, machine learning as a tool for understanding architecture in the brain? So this is a hot topic in neuroscience right now. And various people have indeed suggested that by taking appropriately constrained machine learning systems, you can learn something about the architecture of circuits in the brain. And you know, there's a lot of debate about whether that's true or not, because, you know, the key, I think, is going to be putting in the correct constraints. On a computer, you can do anything, but real neurons have various kinds of constraints. So the question is probably including those constraints in the right way. There's a question from Krishna, which says, why do we have place cells then? If grid encoding is much more efficient, why not just replace place cells with grid cells? So this is an excellent question. So Krishna's question has to do with the fact that in the brain, there are the grid cells that we talked about, which seem to provide a map of space, but there are also place cells. There are also cells in another area of the brain, the hippocampus, which seem to respond at individual locations. Actually, many of them don't respond just a single location. They respond multiple locations, but they certainly do not form these kinds of very geometric grids. So the answer to that question probably has something to do with what you do with these different structures. So the place cells in the hippocampus also seem to respond to other things. So the typical place cell won't just respond to place. It'll respond to, you know, smells and sounds and many other things at the same time. So it's probably more accurate to think about place cells as some kind of context cell, that it represents a certain context for the behavior, and a very prominent context for animal behavior is spatial location. So I think that's part of what's going on, that it's the spatial location becomes kind of a dominant context that's easy to spot when you're talking about that kind of cell. So also place cells in the hippocampus get used for other things. They're used to plan trajectories and things like that. So there's other functions that get performed from that structure. I think that's probably the answer, but the jury is still out and we're still in the process of understanding this. Okay, so then there was a question here from, let me see, go down to the bottom here. Miguel Virasoro asks for questions, for papers that one can read about more about how humans prefer simplicity and robustness. So, so that's something that, well, okay, so that's very new actually. So these questions about simplicity biases are have only been studied in the very recent past. So there's a, let's see, what's the easiest way to provide some references? Maybe I can provide some references offline. I don't know if there's a mechanism to do that, but I will try to do that. Certainly, if you go look at the reference list in the papers which I mentioned, you will find a whole bunch of other references to papers that are talking about these kinds of things. Yeah, so I hope that kind of answered the question. So, okay, so that I did that and I did that. Okay, so going back, so Jiwan Zhao says, is there neurobiological evidence that grid cells firing match that predicted by the N-ary number model? So I tried to provide some evidence there that by saying that the predicted scaling ratio, if you take this N-ary number model, matches the actual scaling ratio that you see, and that one would like further evidence And the further evidence would come about as follows. So there's an experiment we proposed, which is you try to selectively knock out one of these modules. If you do, it's like taking, and if you have a decimal number system, suppose you knock out the hundredths place, then all of your estimates of where you are will be wrong. Well, you won't have information in the hundredths place. Everything else will be correct. So relative to the hundredths place, modulo the hundredths place, you'll be correct. So a piece of evidence would be selective knockout of particular modules. That's a very hard experiment to do, but were you able to do that, you could test this precisely. Hope that answered the question. It's much easier answering questions in person because you can ask them to answer the question. Let me see. Ayush Gautam asks, do we have any analog of horizontal inhibitory cells in artificial neural networks? And the answer is yes. So in artificial neural networks, the weights that you use to connect neurons, units in the network, the other units in the network can be positive or negative. The negative weights are supposed to be kind of morally reflective of the inhibitory cells that appear in the brain. So there's both positive and negative weights. So typical units that you use in a computer, in an artificial network, are much simpler than real neurons. So real neurons, highly nonlinear. They've got multiple kinds of neurotransmitters and ion channels and this, that, and the other. They have feedback. Most artificial neural networks that people work with, even deep learning networks, are sort of feed forward. There's a layer, there's another layer, there's another layer, there's another layer. Whereas most learning in the brain and most networks in the brain are highly fed back. The second layer talks to the first layer. Everything is connected to everything in some complicated way. Personally, I happen to think that the extremely simplistic nature of artificial neural networks also results in the limitations of their learning. So for example, if you look at the state-of-the-art deep learning networks that learn human faces, they need tens of thousands of examples of faces in order to learn them. They're very, very deep. They're 200 layers deep. These are very, very complicated things and huge amounts of data. Whereas people can learn and remember someone else's face, essentially after looking at them once. It's sort of one-shot learning. And what's more, observe that the number of synapses between the eye and the fusiform face area, basically the area of the brain where you can recognize faces, like 6 synapses. It's not 200 layers deep. So personally, I think the extreme simplicity of neural network architectures is part of the reason why, and the lack of feedback, top-down feedback, is part of the reason why they are basically much, much less efficient than real networks in the real brain. Okay. Here's an interesting question. Here are 2 questions from Tanya Jaiswal. Do the brains of different organisms, all organisms, follow a particular number system, or it varies? So data on these kinds of grid cells is limited to a few species right now. We have information from rats and mice, a little bit from humans, from MRI studies and fMRI studies, and a little bit from bats. And in humans and rats, it appears to be similar. In bats, there's some 3D-ness involving the flight of the bats. That's still very much a hot topic under current study, so still up in the air. So let me answer that. Then there's another question from the same person, Tanya Jaiswal, saying, how can this be used to define dreams? Now, there's an interesting question. Dreams. So, it's sort of unclear exactly what dreams are, but there has been much speculation in neuroscience that dreams involve spontaneous activity of networks of neurons in the brain that are partially used to consolidate memory and to imagine solutions to things. So for example, there have been studies done with rats that have run mazes. And so they'll run the maze and you'll record from a whole bunch of neurons. You'll see them firing. Let's say you see some particular neuron firing when the rat reaches a particular point in the maze and turns left. And then another neuron firing when the rat reaches the next point in the maze and turns right. So then you'll find that during sleep, when the rat's asleep, you'll find playback in the hippocampus, which reflects that sequence of firing that you saw when it was awake. So we infer from that probably that there's some kind of dream going on, in which it's playing back again and again the trajectory it took, and that this is somehow involved with memory consolidation and the strengthening of synapses. Very interesting kind of thing to think about. There's also work done, I think, by Google a couple years ago, in which they caused well, maybe it's a poetic way of talking, but they took a deep network that had learned to recognize, I don't know, lots of images or pictures or paintings or something, and they caused it to dream. Namely, instead of using it to recognize things, you feed it input, and at the other end you recognize what it is. They ran it the other way to sort of generate output, and it produced a whole bunch of hauntingly beautiful things that looked like, I don't know, interesting paintings of some kind. So what that tells us precisely is unclear, but there you go. So let me see. So that's another question. So I think we're at 1 hour and 45 minutes. I'm not sure if I'm supposed to stop. I'm happy to keep going to answer a few more questions. Should I do that?
Vijay, you tell me when you would like to stop.
Okay, so let's see. Let me just take a look at this. I'm just going through the questions to see what's left.
You answered some questions, but you didn't mark them as answered, so.
Yes,
Yeah.
so I am trying to mark off some of these things. Shuyin Jianwu has a very interesting question, saying, would the optimal ratio of off and on be different for nocturnal and diurnal animals? For humans, it seems that we do most of our visual processing when the sun's up. Excellent question. So a very good way of testing the kind of theory you talked about is to have animals that are living in some sort of modified visual environments. And then you would want to take those modified visual environments, study their visual statistics, and then make a similar prediction and see does it match. That would be a good test, right? These kinds of studies haven't been done so much with the nocturnal animals, so we don't have a detailed piece of information about the relative ratios of off and on, and we also don't have detailed information about the structure of light in quite the same way. Nocturnal environments are fundamentally different because there's so little light in true nocturnal environments that you cannot even use the same circuits to detect the same kind of visual features that you would normally in daylight. So so the problem has to be recast and kind of done in a kind of different way. One thing that we're doing explicitly right now with the collaborator Davide Zoccolan in Sissa in Italy is to raise animals in modified visual environments that reverse the bright to dark ratios, for example, and then try to ask, does the structure of the responses in the brain change in a way as predicted by theory? It won't change in the retina because the retina happens to be wired in utero. So when the animal is born, it has its off to on ratio in the retina fixed. But that's not true of cortex. Things change very much in cortex as the animal opens its eyes. So we're trying to make some predictions using a similar theory for how the response properties of cortical neurons will change if you are in an environment, let's say, with more bright spots than dark spots. And well, we hope to tell you what happens in some time. Soon, we hope. We'll see. Okay. I've marked a few more as done. There's a question, what advice do you have for physics majors who are interested in doing research work where physics is applied to biology? So I think the first and most important thing is to be really good at physics, because the techniques you learn from all branches of physics are going to be useful. So in particular, statistical physics is of course super useful, but so is dynamical systems, and it's a good idea. For example, there are lots of things that you might think are not relevant to biology that are relevant. So for example, you might think general relativity is obviously irrelevant. Yes, general relativity of Einstein per se is not, curvature of space and time is not directly relevant. On the other hand, it's by studying general relativity that I learned all the tools of differential geometry that allowed me to think personally about probability distributions as being sort of curved manifolds in the space of distributions. And then basically, the paper that I gave you guys to read from 1997 was actually my first paper as a graduate student. And I wrote that paper because at that time I was studying general relativity and I knew these techniques, so I applied them to probability distributions. And that's come back a circle. And right now we're doing experiments to test the predictions of that theory. So basically learn everything. It's all going to be useful. Regarding biology, you know, this also having deep knowledge of biology is also useful. The challenge there is, you know, the living world is vast, complicated, and exceedingly diverse. So in my own case, it's been a matter of I have to be interested in a question, and then once I know that I'm interested in some question, sort of dig into it. I ask my biology friends to sort of give me papers to read and dig out from there, and then expand into knowing enough in that area to be able to say meaningful things. But so I think this, but you know, it's that's knowledge you can learn in biology like that. But the techniques, the calculational techniques, and the methods of and the habits of thought developed by physics are really, really useful. So those should be developed early. I hope I answered that question. Okay, and okay. Okay. So let me finish up here. Right. So Azim Khan writes something about the bats using echolocation rather than the eyes. Absolutely. So nothing I said depended on the use of a particular sense to find out to arrange yourself. In fact, a lot of the formation of grid cells in animals is not even based. There's a significant component of the formation of these sort of mental maps, or the cognitive maps, that's not even based on sensory input, vision, sound, or anything else. There's a significant portion of it that's based on path integration, where what you do is you use your knowledge of the use of your own muscles to integrate up to your total distance. So it's possible to do that. And in fact, for example, there are a certain kind of desert ant, you know, starts at its nest and will do some sort of crazy path looking for food. And when it gets to the food, it turns around and aims straight back at its home nest and goes straight there. The way it does this, apparently, is that there are circuits in its brain that add up the vector direction it's going in. It knows the velocity because, you know, it's generating its own velocity, and it knows the direction. So it adds up the vectors, and at the end, it can calculate the vector direction back home. So that's an example of doing path integration. So grid cells are thought to, and there's significant evidence for this, partly be created by path integration. So bats are presumably doing that too. Of course, that'll interact with things like visual sensing and echolocation and other kinds of sensors that you use to sort of stabilize and control your spatial map. Okay, so I think I've answered lots of questions. That isn't 100% of them, but I'm gonna look for one more here. I think that's most of them. So I think since I have answered most of them, shall we call this victory?
Okay, we shall. Thank you very much, Vijay.
Thank you.
Okay, wonderful. And thank you very much. Bye now.
Bye.